Hey, what's up, everybody? This is Mr. Conway, and this is going to be a continuation of the unit sensation and perception, and we're going to be focused mainly on the eye during this video. Now, hopefully, I can get through the eye and the retina, but just in case, I'm probably plan on having another video just solely on the retina. Okay, so let's get started. I'm looking at printed page 124. 4.2 on the vision. I'm looking right at the ebook, okay? And one of the first terms it gives you is transduce, okay? Now, you're going to see it in the form transduction. And this is a really important concept, not just for vision, but for the whole unit. Okay, when you look at transduce, it has parentheses and it says to transform. That's a really good mnemonic to use for this word. So what's happening is your brain, this is how I like to explain it, your brain can only understand one language. Okay, what language do you think that is? If you said English, sorry, no. And it's not Spanish either. The brain can only interpret electrical signals. So your brain is very, very electrical. Those neurons, it's an electrical process, okay? So the brain can't actually interpret these like light waves that you're going to read about in a second, or sound waves, or even the sense. Anything that we pick up with our five senses must be converted into an electrical signal so that the brain can then read it and interpret it. And that process of converting it from a wave, like a light wave or a sound wave, converting that into an electrical impulse is called transduction. So that's going to be a really big word that you're going to need to understand. And let's go to the next page. What are we transducing now? What are we transforming? So we see a picture of the light spectrum. If anybody's Pink Floyd fan, this is like the cover of Dark Side of the Moon. Probably not. Okay. All right, let's go to the bottom of, I'm using the ebook, printed page 124, the very bottom, figure 4.9. All right, this is going to be big. You're going to have to know the difference between wavelengths and amplitudes, okay? Both for vision, for light waves and for sound waves, okay? So when we're looking at that figure 4.9, okay, the left part of it is going to be talking about wavelengths. So how far the waves are, or the top of the wave to the other top. How far is that? Okay, that's called a wavelength. You can see the little arrows, all right? If it has a high frequency, that means that the waves are repeating very, very often. That means that we have a short wavelength. If it's a high frequency, short wavelength, okay, that's the same thing. That's going to give us a bluish color, all right? If we have a very long wavelength, that's a low frequency. That means that the frequency of the waves are not happening as much. That's going to be more of a reddish color, okay? You're not going to have to pick out what wave is what color. That's not the point. But what you will have to understand is that frequency or wavelength is going to determine the color. So a high frequency, that means very short wavelength, lots of them, that's going to be blue, long, red. Now, don't have to really memorize that, but wherever those wavelengths are, maybe it's a medium wavelength, that's going to be somewhere in the middle of that color spectrum. Maybe like a green, maybe like a yellow, something like that. So just know that the wavelength determines the color, okay? Another word for color you're going to see is HUE, hue. What is the hue? Okay, that's the same thing as color. So know that. The other thing on the right side of 4.9, you have to know the amplitude. That's going to be how tall the wavelength is from the bottom of the wave, from its lowest point to its highest point. That's called the amplitude, anywhere in between, okay? Again, you're not going to need to know exactly how bright it is, but you just need to know that that does determine the brightness, or another word for that is the intensity of the color. So just if it's a very tall amplitude, it's going to be a very bright color. So those are going to be like your neon colors, perhaps. And then if it's a low amplitude, that's going to be like this color shirt, right? It's really dull, or like a navy, or something like a really dark color, okay? So amplitude determines the intensity or the brightness. If it's a high amplitude, it's going to be a very bright color. If it's low, this can be a very dull color. And then wavelength determines what the color actually is, okay? You're going to need to know that. All right, let's keep moving through. Now we're actually looking at the eye, okay? So you should see at figure 4.10 the structure of the eye, okay? And it gives you about seven or eight different terms that you need to know, all right? Now, all of these are of some importance, okay? But the biggest one is going to be the retina. Okay, the retina, they're actually going to focus on that here in a second. So what I would suggest, I'm not going to spend a lot of time going through these other ones. You can do that on your own. You may get a couple questions on what the function of the iris or the lens or the pupil. It's kind of low priority. It's really, you're going to get some quiz questions on it, so you're going to want to know it for sure, but I'm going to spend the rest of this time of the video talking about the retina, okay? So let's go through it. Now, something you're going to want to pay attention to with, you do want to check out accommodation though. Accommodation is how the lens changes its shape to focus on things that are far away or close up. And we'll probably do something in class with that. That's why, that's what determines what your focus is and why things are clear and why some things are not. But let's go ahead and scroll down. Let's jump into the retina, okay? Figure 4.11 is something that you're going to definitely want to be familiar with, okay? This is going to be a close-up of the retina. Now, when I first started learning this stuff, I thought that the retina was like in the front of the eye or something. I thought it was like some structure. And it's really strange the way that it's set up, okay? If you actually kind of scroll back up to that figure 4.10, the retina is this back layer of the eye. Okay, it's like the back, it goes all the way down, starting at the top of it, and it goes all the way down to the bottom. It goes kind of stops at the optic nerve, and we'll talk about what that is. But if you go down to 4.11, this is what the retina looks like, and this goes along the entire kind of back of the eye. It's like a thin surface, okay? So I thought that was kind of weird because I always thought that the retina is just being like a condensed, like, part of the eye, like the lens or something, but it's actually just this really long layer that goes around the eye, okay? And here's something, the process of transduction actually occurs in the retina, in this retina right here. So, and look at this process in figure 4.11. So the light, when it comes through your eyeball, you can actually see that being modeled right here. So the light comes in, you can see the arrows going down, okay? When the light comes in, it goes all the way to the back of the retina, all right? The neural impulse, the light goes to the back of the retina, and what it does is it stimulates, you can see on the picture, these red and green neurons. These are rods and cones, okay? These are going to be what helps you see in the dark and what helps you see color. So rods and cones are going to be the first thing that's stimulated by the light, okay? Good mnemonic is cones help you see in color. Your rods are going to help you see in your night vision in black and white. So we actually have way more rods than we do cones. Okay, so once these rods and cones are stimulated, what happens is they actually pass a neural impulse onto the bipolar cells. Okay, not much here with the bipolar cells we're going to talk about, except that they're right after the rods and cones. There's going to be a question on your test that talks about the order of a neural impulse in the retina. So it starts with the rods and cones, then it goes to the bipolar cells. A good mnemonic to know that bipolar cells is second is because bi means two. So the bipolar cells comes after the rods and cones. It comes second, okay? And all that's going to do is kind of be the middleman. Once the bipolar cells are stimulated, then the ganglion cells are going to be stimulated, okay? Another thing you could remember, bipolar, bi meaning two, meaning it's got something on either side, meaning it's in the middle. So the light comes into the retina, stimulates the rods and cones. They stimulate the bipolar cells, which stimulate the ganglion cells, okay? The ganglion cells is pretty much the last stop, because what happens is the tails of those neurons actually converge, meaning they come together and they form the optic nerve. You've probably heard the optic nerve before. In this spot right here on the, I know you can't see what I'm pointing at, but if you're looking at figure 4.11, you see these black arrows going down, and it says to the brain's visual cortex via the thalamus. So what happens is this optic nerve carries this, it's now a neural impulse. So it came into the eye as a light wave, but through the process of transduction, those rods and cones, the bipolar cells, and the ganglion cells have transformed it into an electrical impulse. Now the brain can interpret that as something, as vision, as what we see. So that neural impulse goes down the optic nerve, and then it actually gets sent to the thalamus, which then sends it to the visual cortex, which is in the occipital lobe in the back of the brain. And that's actually how we see. Okay? We're going to, you can try to test your blind spot here with 4.12, figure 4.12, but if you notice right there where that optic nerve leaves the eye, there's no neurons, there's no feature detectors, there's nothing there. So we actually have a spot that we can't see when light reflects and it hits that blind spot. There's nothing to detect it. So what's cool is our brain actually fills in that gap and we don't actually notice that it's a blind spot. And I'm going to show you how you can find your blind spot to where stuff will disappear. It's pretty cool. So we'll do that in class. One thing I do want to talk about is the fovea. This is going to be the central focal point. That's what I'm reading right out the definition. In the retina, which is where there's a high density of rods and cones. So your fovea, if you go back up to figure 4.10, that's actually going to be the central point of focus. That's going to be where your lens is going to try to focus all of the light waves onto the fovea so that we can get that true clarity. Okay. Let's see. Let's go on to the next page here. Okay. Now, I think what I'm going to do here is I'm going to finish up with visual information processing and then we'll do something on color vision. So look at this figure 4.13. I'm on printed page 128 if you need to pause. This is going to be, this explains that contralateral control that we talked about with the, when we're talking about split brain procedures. Now you can see how the left eye will send information to the right occipital lobe, and the left eye will send information to the right occipital lobe, and the right eye will send it to the left occipital lobe. So that is actually what's happening. And although we saw in split brain patients that the corpus callosum was severed, and although the brain wasn't communicating, we did know that the visual information is still being sent to the respective lobes. All right. Now, the brain has something cool inside the occipital lobes, and those are called feature detectors. I want you to check out feature detectors. These are nerve cells that we have that respond to angles or movement. So we are more likely to look at a room and we can tell that there are angles that come together to make things that we're familiar with, like a room or a chair or even faces. So feature detectors are they're kind of like clusters of special types of nerve cells that help us recognize things. And those are also in the occipital lobe. And the last thing I wanted to touch on before I run out of time is parallel processing. This is a pretty popular word, but it's really not that complicated. There's a picture 4.15. This just says that the brain does a lot of things at once. We don't do step-by-step processing like a computer. What happens is when we look at things, we assess the color, the motion, the form, the depth all at once. We don't have to sit there and go, oh, that looks like, that's the shape of a bird, that's the depth of a bird. Then we have to do the color, is that a bird? And then, no, we do it all at once. Our brain, and that's what kind of parallel means, it doesn't cross over, it kind of goes on infinitely with something next to it. So what happens is we can parallel process multiple things at once without it kind of crossing over. So I would definitely check out parallel processing. We can recognize things, and it's going to be something pretty crucial to our visual processing. Okay, so I am going to do another video on color vision. So thank you. Click subscribe.